In our previous sessions, we have seen the classification of vitamins, that is fat-soluble vitamin and water-soluble vitamin. Today, we shall look at one of the fat-soluble vitamin, which is vitamin A. Basically, there are three important forms of vitamin A, that is retinol, retinal, and retinoic acid. All the three compounds contain as common structural unit a trimethyl cyclohexanyl ring, an all trans configurated polyene chain with 4 double bonds, and they are crystalline substances with limited stability. The trimethyl cyclohexanyl ring is beta-ionone. Let's look at the forms of vitamin A. Vitamin A occurs in nature in different forms. The usual form is vitamin A1, which predominates except in freshwater fish. In them, another form, namely vitamin A2, is present. Neovitamin A is a stereoisomer of vitamin A1 and it has about 70 to 80% of the biological activity of vitamin A1. So neovitamin is the stereoisomer of vitamin A1. Moving on to the difference between the two vitamins, that is vitamin A1 and vitamin A2. Vitamin A1 is found predominantly in major species of animals, whereas vitamin A2 is found only in freshwater fish, liver, and other tissues. Vitamin A1 has only one double bond present in the beta-ionone ring, whereas vitamin A2 has two double bonds in its beta-ionone ring. The additional double bond is present between the carbon 3 and carbon 4. Vitamin A1 is more potent in its activity when compared to vitamin A2. Vitamin A1 can be obtained from carotenes, whereas vitamin A2 cannot be obtained from carotenes. The dietary sources and daily requirements. The major animal sources are liver oil, butter, milk, cheese, and egg yolk. Other plant sources in the form of provitamin carotene are tomatoes, carrots, carotene, green yellow vegetables, spinach, fruits such as mangoes, papayas, corn, sweet potatoes, and algae called as spirulina also is rich and a good source of vitamin A. An adult male and female require about 3000 IU per day of vitamin A. However, a recommended allowance is around 5000 IU per day. It is higher in growing children, pregnant women, and lactating mothers. The requirement is also higher in hepatic diseases. Let's move on and see how the absorption of vitamins take place and also see how the vitamins are stored, specifically vitamin A. Vitamin A and its carotene precursors are absorbed in the small intestine. It is believed that the presence of tocopherols, that is vitamin E, and other antioxidants protect them against oxidation and destruction in the intestinal lumen. Dietary vitamin A is chiefly in ester form, which is hydrolyzed by cholesterol esterase into fatty acids and free vitamin A. The free retinol is then absorbed and undergoes re-esterification in the intestinal epithelial cells. It is stored in the liver as retinyl ester, normally as retinyl palmitate. Now we'll look at how the vitamin A is transported. Conversion of carotenes to retinol occurs in intestinal epithelial cells in few animals, although liver also participates in this conversion. However, in men, liver is the only organ where carotenes are converted to vitamin A. Retinol is transported in the blood in association with a specific retinol binding protein RBP. There is another retinoic acid binding protein RABP specific for retinoic acid. About 95% of vitamin A is stored as its ester mainly as palmitate in liver. It is released in the plasma as and when required. About 10 to 20 milligrams of vitamin A is present per 100 grams of liver. So about 10 to 20 milligrams of vitamin A is present per 100 grams of liver. It is 10 to 20 milligrams. The normal blood level of vitamin A is found to be 18 to 60 microgram per deciliter, and that of carotenoids is 100 to 1000 microgram per deciliter.